Hello and welcome back to Microbial Concepts. So today we are going to discuss about bacterial transduction and conjugation. Already a video is uploaded on bacterial transformation. It is there on my channel. You can watch it out. And just we are going to discuss here transduction and conjugation. So we have already studied that in transformation video as well, that vertical transmission is where generally the way our bacteria they divide by binary fission, and the genetic material is also copied during cell division and it is passed to the offsprings. Natural way of passing the genetic material to the offsprings is vertical transmission. Horizontal transmission is where mutations are transferred from one bacteria to another. Okay, so this is not seen in each and every bacterial cell or species. So the frequency of horizontal transmission is different, and even the frequency of mutations which are getting transferred to horizontal transmission is different. Now there are three different types of horizontal transmission, of which we have covered transformation, and just transduction and conjugation is what we are going to focus. So let's start with transduction. So transduction is the transfer of bacterial DNA from a donor to a recipient bacterium via a virus particle. Now in case of transformation, there was no any other agent like a virus particle was used. Okay, only the competent cells, that means the cells which are altered for the cell wall permeability, they were able to take up free bacterial DNA which is present in the surrounding. Okay, in case of transduction, virus particles are one which carries out the transfer of bacterial DNA. The virus particle that infects bacteria, they are generally bacteriophage or phage, and the phages used for the transfer of DNA, they are called as transfusing phages. Not all phages are transducing phages. The process of transduction can transfer DNA regions of 10 to hundreds of kilobases. In case of transformation, it was 1 to 10 of kilobases. Now phages have the high specificity for the cell surface receptors. So transduction has the narrowest host range for DNA transfer among the methods or other methods for bacterial genetic exchange. Transduction involves the carrying over of DNA from one organism to another by an intermediate agent, that is bacteriophage. So you can easily remember that in case of transduction, there is a agent that plays an important role in transfer of bacterial DNA, that is a virus particle known as bacteriophage. In case of transformation, cell wall permeability is altered and bacterial cells are made competent so that they can take up the free DNA on their own. Now transduction is a standard process employed by many molecular biologists to introduce a foreign gene into a host cell genome. Okay, so here the specificity is taken into consideration of the virus particle towards the host cell. Now transduction has an advantage over conjugation. Now conjugation is the third uh, method where direct cell to cell contact is made. Okay, cell to cell contact in between a donor cell and a recipient cell. Okay, now the advantage is that transduction does not require physical contact between the cell donating the DNA and receiving the DNA. Okay, so this is the difference between transformation, transduction and conjugation. Similarly, the process of transduction is resistant to DNase enzyme, while the transformation process is susceptible to the DNase enzyme. So yes, so when you are performing the transformation experiment, you have to take care of DNase enzyme as they degrade your DNA. So the bacterial transduction principle. The principle of transduction is based on the mechanism of infection of the bacteriophage. In transduction, the bacterial donor DNA is incorporated into bacteriophage either through lytic or lysogenic cycle, and after the bacterial DNA is incorporated into the phage, new phages are released from the bacterial cell. Okay. Now, after the bacterial DNA is incorporated into the phages, it will replicate when it infects a bacterial cell, and new phages are released from that particular bacterial cell. That's what the point is here. I'm trying to explain. Now, these phages, they infect bacterial host cell. Phages attach to the specific bacterial cell surface receptor, and they inject the DNA containing the donor DNA into the cytoplasm of the host bacterial cell. Now, depending on the phage, the DNA integrates into the bacterial genome, replicates in the cytoplasm as a plasmid, or it can replicate immediately, producing phage progeny. Now, this depends on the type of phage. It can integrate the DNA to the bacterial genome, or if it is plasmid, then it can replicate in the cytoplasm, or it can start replicating immediately to produce phage progeny. Now you can see a diagram here. So first you will see this is the bacterial DNA with chromosomal DNA. So bacterial cell with chromosomal DNA, it has a phage which has attached to the host cell as it is specific to the bacterial cell. It injects the bacterial DNA, or the DNA that is to be injected. Okay, so the phage host cell, that is donor cell, is first infected by the phage, during which the phage DNA enters the cytoplasm of the bacteria. So you can see the DNA is entered into the cytoplasm. During the lytic cycle of viral replication, the phage DNA along with the bacterial chromosome is broken down into the smaller pieces. Now some part of the bacterial chromosome is then packaged into one of the viral capsids when that is released by lysis of bacterium. So you can see the green color is the bacterial DNA and red color is the viral DNA. So as you can see, the bacterial chromosome along with the phage DNA is broken down into smaller pieces. So the bacterial chromosome is also getting packaged into the some of the viral capsids. Okay, and it is released when the bacterial cell lyses. The transducing phase, with the bacterial chromosome now infects a second bacterium, that is recipient bacterium, and the donor DNA now enters the cytoplasm of the second bacterium. Okay, so now you can see here this particular phage particle has a bacterial DNA, so it can infect another bacterial cell and it can inject the bacterial DNA into it. In presence of a host recombinase Rec-A, the donor DNA recombines with the homologous DNA of the bacterial recipient to generate stable transductants. Okay, so now once the bacterial DNA is released into the cytoplasm of the cell in presence of host recombinase Rec-A gene, the donor DNA, it recombines with the homologous DNA from the chromosomal DNA of bacteria, and it gets incorporated or integrated into it. Now this particular cell is termed as bacterial transductant, as it has a new piece of, or the foreign piece of DNA that has got integrated into its chromosomal DNA. Now there are types of transduction. So based on how the DNA is packaged within the viral particle, there are two types of transductions. Generalized transduction is the first one. So here what happens in generalized transduction, phage mistakenly packages DNA of bacteria instead of its own phage DNA during phage assembly. Now this results in an infectious virus particle containing bacterial DNA. Okay, means the phage or the virus particle, it is now having bacterial DNA but not the viral DNA, but one that can no longer replicate in the bacterium due to the loss of all the phage DNA. So this particular DNA cannot replicate in the bacterium on its own as it has lost the phage DNA. Now the phage particle then attaches to the bacterial cell surface receptor and injects the packaged DNA into cytoplasm. If the bacterial DNA in the phage is from the bacterial chromosome, then DNA recombines with the homologous DNA of the bacterial recipient to generate stable transductants. This process requires host recombinase, that is REC-A. However, studies have indicated that the majority of transduced DNA is not stably integrated into the bacterial genome, but rather remains extrachromosomal. Generalized transduction is used for mapping genes, mutagenesis, transferring plasmids and transposons, and determining whether different different genera of bacteria have homologous genes or not. So these are the applications of generalized transduction. Another type is specialized transduction. So here what happens? Phage undergoes lysogeny or lysogenic cycle, usually at specific location in the bacterial genome called attachment site. During this process, phage genome usually integrates into bacterial chromosome as virus replication is repressed during lysogeny. So the phage DNA integrates into bacterial DNA as virus replication is repressed during lysogenic cycle. The phage genome then excises from the bacterial genome, and due to excision and recombination, adjacent bacterial genes are also excised. Okay, so some pieces of bacterial DNA, they are also excised, means they are taken out or they are cut off with the phage genome. Unlike a generalized transducing phage, specialized transducing phage, they contains both bacterial and phage DNA or genes, not the complete DNA, bacterial and phage genes. During the subsequent infection, a newly acquired gene is inserted into the bacterial genome along with the phage DNA to form a new round of lysogeny. So now this will be a new lysogenic cycle as it contains both the genes, viral and bacterial, when it infects a subsequent bacterial cell. Now, specialized transduction is independent of host homologous recombinase and REC-A, but requires phage integrase. So this is the difference between generalized transduction and specialized transduction. Specialized transduction, it requires phage integrase, and generalized transduction requires homologous recombinase, uh, that is REC-A. Okay. Now, specialized transduction is instrumental in the isolation of genes in molecular biology and in the discovery of insertion elements, which often serve as attachment site for phage DNA integration. So that was about transduction. Now about conjugation. So conjugation is transfer of plasmid or other self-transmissible DNA elements, or sometimes chromosomal DNA, from a donor cell to a recipient cell via direct contact, usually mediated by conjugation pilus or sex pilus or pilus. Okay. Now recipients of DNA transferred by conjugation, they are called as transconjugates. The process of conjugation can transfer DNA regions of hundreds to thousands of kilobases and has the broadest host range for DNA transfer amongst the other methods of bacterial exchange. Conjugation occurs in between many species of bacteria, including gram-negative as well as gram-positive bacteria. Although numerous examples of conjugative plasmid exist, conjugation involving the F plasmid is most common. Now you can see here, this is the donor cell, this is the recipient cell. The donor cell has a shuttle plasmid. So this plasmid will get transferred to the recipient cell. So how this happens? Direct contact between donor and recipient cell is made. Then the copy of a plasmid is also made by using relaxosomes and transferosomes, and thus a similar copy of plasmid is transferred into the recipient cell. Now, principle of bacterial conjugation. The process of bacterial conjugation is based on principle that plasmids or any genetic material is transferred from donor cell to recipient through close physical contact. Of all the conjugative plasmids, F plasmid, which is also known as fertility plasmid of E. coli, was discovered first and is one of the best studied fertility plasmids. The F plasmid is present in 1 or 2 copies per cell and is very large. It has around 100 kilobases of the sequence. Now E. coli, which is harboring or which has this F plasmid, is referred as donor, that is F plus or male cell, and E. coli, which is lacking this F plasmid, is referred as F minus or female cell. Only donor cells are capable of transferring F plasmids to the recipient cell. So for transfer of F plasmid from donor to recipient, intimate contact between cells resulting in mating pair or mating pair formation is required. The transfer of genetic material is then brought by membrane fusion of the two cells by the action of different enzymes, and following the membrane fusion, the replication of donor DNA occurs, or as we saw in the previous slide, there was a plasmid, so the replication of plasmid is made, a copy is made, and then it is transferred to a recipient cell. The following process occurs during the transfer of F plasmid in E. coli by conjugation. So you can see this is the donor F cell, male cell, which has the fertility plasmid, and the recipient cell which does not have a F plasmid. So you can see here a copy is being made of the F plasmid and then it is transferred to the recipient cell. So F plasmid contains TRA loci, which locus, which includes the pyline gene. Okay, so this gene along with some regulatory proteins results in the formation of pili on the F plus cell surface. The protein present in the pili or attach themselves on the F minus cell surface. The pili are responsible for making contact between the cells. Okay, so pilin retracts, mating pair forms. So you can see here the cells are fusing here, but the transfer of plasmid does not occur through the pili. The TRAD enzyme located at the base of the pilus, it initiates the membrane fusion. So now you can see here membrane fusion has taken place. Once the conjugation is initiated, enzyme relaxes, creates a nick in the conjugative plasmid at the ORIT, that is origin T. Okay, so this is how you can see here enzyme relaxes has created a nick in the conjugative plasmid and at the ORIT, that is origin T. Okay, now the nick strand then unwinds and is transferred to the recipient cell in 5' to 3' direction. The complementary strand is synthesized in both these cells. As there is just one strand of the plasmid in recipient and donor cell, the complementary strand has to be synthesized by both the cells, and thus both the donor and recipient cell are now F plus, that is they have fertility plasmid present in both. In certain F plus bacterial cells, the F element infrequently becomes associated with the main bacterial chromosome. This is in some cases. In such a way, a copy of chromosome instead is transferred through the conjugation tube from the donor to the recipient. Okay, so this happens about once in every 10,000 F plus cells. In the insertion process, the circular F element, it breaks at a particular point and becomes a linear segment of bacterial chromosome. An F plus cells that carries such an integrated F element is also known as HFR cell. Now HFR stands for high frequency of recombination. The integrated F element of HFR, that is high frequency recombination cells, is ordinarily replicated passively along with the bacterial chromosome, and in this way is transmitted from one HFR generation to the next. So this is the once in every 10,000 cells case. Okay, so this was about conjugation, and we have covered transformation and transduction as well. So you can see here from a dead cell from which a free DNA fragment is released into environment, can be taken up by transformation by a bacterial cell which is competent, then it can be injected to another specific uh, bacterial cell through transduction, where Phage particles carries such kind of DNA segments, or it can be conjugated, means the fertility plasmid is the example here, which is conjugated or it is transferred from one bacterial cell to another through direct contact. So these are the all three methods of horizontal transmission. So I hope these concepts are clear. Do like my videos, do share my videos, and do subscribe to my channel. Thank you.